underway and be done with it, okay? I know, and it's Friday, good, even though your whole weekend is ruined because of finals week. I know, it sucks. Okay, so what we're going to do today is two things. Obviously, we have a little bit of stuff that still needs to be wrapped up. We only got to fertilization yesterday and early embryo development, okay? So again, for female reproductive physiology, for the final exam, really just keep track of these hormones, okay? And kind of think about the fact that, start with the ovary, okay? Where are we? Are we in the follicular phase or the luteal phase? It causes lots of hormone secretion, and that's going to impact either the brain or the uterus, okay? And if you keep that line of thought constantly going, it hopefully makes somewhat sense, okay? So after fertilization, we kind of recognize maybe too that, wow, it's still kind of a miracle that it does happen, even though initially we thought 200 million sperm is way too much. But by the time we recognize how few actually get to the egg and then all the other stuff that still has to happen, it's still kind of amazing that as humans, we're still populating the globe as we do, and we're doing a very good job with it, okay? Okay, so but from the embryo part of things, I really want you to recognize certain terms like zygote, okay, morula, blastocyst, okay. When we talked about fertilization, I want you to understand what happens, the two things that happen as soon as a sperm fuses with an egg, okay. Terms like capacitation, acrosome reaction, what do they mean? When do they happen? Where do they happen? Okay, so those are all important things for the exam, okay. Okay, so we talked about fertilization. We know where it happens. It's in the fallopian tube. And we recognize the fact that the sperm and an egg have fused. We now start as a zygote, and the zygote starts dividing. We get to a 2-cell, 4-cell. And as that embryo continuously divides, what happens is it starts moving from the fallopian tube towards the uterus. Okay? And when it does so, okay, it's going to get at some point into the uterus after about a week, and this structure is called the blastocyst. And the blastocyst is an embryo that had two groups of cells. One single cell layer on the outside, called the trophectoderm, that's going to be the placenta, and a group of cells on the inside, which is the embryo, really. And these cells are embryonic stem cells. Okay? So these two groups of cells, embryonic stem cells, those cells can give rise to everything that makes up the body, but not a placenta. And we have trophoblast cells, or trophectoderm. They have stem cells too. And these trophoblast stem cells can give rise to all the different cell types that make up a placenta. And we'll talk about a placenta in a minute. Okay, okay, so this figure here kind of highlights all the areas in the uterus where we can have implantation. So does it look like there's really any preference? Not really, right? I mean, it could be left, right, on the top. It doesn't matter, okay? If we look at the structure of the blastocyst again, here's the inner cell mass. So underneath it is a single layer of trophoblast cells. And as soon as it attaches to the endometrium, proliferation, penetrate, invade, and you get implantation, okay? Like I said, it's kind of like a cancer, okay? This is a cartoon picture. The green cells here are the trophoblast cells. And here you see invasion and migration. And they really invade quite deep into the uterine lining, okay, the endometrium. They can go, it is very extensive, okay. Now, when we talk about the placenta, I'll do that later on. When we talk about the placenta, recognize too that this is quite a unique structure because that placenta is only present in women for 9 months. That's it. Okay, so we'll talk about the placenta, a little bit about its structure and blood flow through the placenta. Let's see. Okay, implantation. Where or when does this happen during the monthly cycle? Where are we in the monthly cycle? If we assume that fertilization has occurred and embryogenesis has occurred. Luteal phase, okay. So in the ovary, we have a corpus luteum. And the corpus luteum does what? Estrogen, progesterone. And this was very important because it does what? Yeah, the endometrial lining, right? Really thick. That actually allows the embryo to attach, right? So if this really happens, okay, this process, or this one, What needs to happen? Because what's going to happen normally during the monthly cycle, during the luteal phase, after a little while? Yeah, but what happens in the ovary? In the ovary. What?
Follicles don't produce eggs?
Yeah, they don't produce eggs, but what more specifically? Related to estrogen and progesterone. Why? What happens to the corpus luteum? It dies off. Remember how long the corpus luteum was around for, approximately? 10, 11 days? So that picture right here is after about 7 days, maybe 8 days. Okay? So what needs to happen right now? Yeah, so we keep going there, but what basically the ovary needs to be told something. What cannot happen in the ovary that normally does happen if this is not occurring? Yeah, so do we still want the corpus luteum to die after 10 days? Oh, there we go. Okay, we'll get there. I know it's Friday. I'm just as tired as you are. So basically, if this happens, okay, one of the first things that embryo has to do once it attaches to the endometrium and starts implanting, it has to tell mom or the ovary, keep that corpus luteum alive, stop killing it. Okay? And basically, that is the process of maternal recognition of pregnancy. Maternal recognition of pregnancy. Different animal species do it different ways. Since this is human physiology, we're just going to focus on humans. Okay? So what happens is after the embryo has attached, this embryo is going to tell mom, I'm pregnant. Okay? So I'm here. Don't kill the corpus luteum. Maternal recognition of pregnancy. The hormone responsible for this is human chorionic gonadotropin, or HCG. Okay? So in fact, this embryo structure, the blastocyst that's on this figure right here, again, the trophectoderm on the outside, inner cell mass on the inside, it is actually the trophoblast cells, or the trophectoderm, that secretes that hormone, human chorionic gonadotropin. Okay? So the hormone is released, it goes through the endometrial wall, goes into the bloodstream, goes to the ovary, and protects the corpus luteum and keeps it around. And we all know that that's really important because we want all that estrogen and progesterone to maintain the endometrial lining. If we do not have enough estrogen and progesterone, that endometrial lining sloughs off, including that embryo, and we start a new cycle. Okay? Does that make sense? So human chorionic gonadotropin, another hormone, but it's extremely similar to a hormone we already talked about. It is almost identical to luteinizing hormone. Okay? The only difference is that human chorionic gonadotropin is more glycosylated. So it is a little bit more stable in blood. It stays around a little longer. Okay? But for all intents and purposes, it's kind of like LH. Okay? So human chorionic gonadotropin. This figure here kind of shows what's going to happen if that is around. Okay? You maintain the corpus luteum. Instead of progesterone levels dropping, estrogen levels dropping, we keep that around, and we sustain progesterone levels. Make sense? So, how many of you are familiar with a pregnancy test? It's kind of a loaded question because that... Or at least, how many of you have heard of the fact that there are pregnancy tests? Ah, there we go. That's probably a safer question to ask. So how does that work? Yeah, you pee on it. And presumably, the women pee on it. If a man pees on it, it's not really going to do anything. Okay? What are you looking for? HCG, exactly. Basically, the pregnancy test, that strip, is loaded with antibodies for HCG, and it specifically detects HCG in urine. Okay? I know Dr. Warren talked about the kidney, and you guys all aced the renal section on that exam. And so you also know the blood gets filtered. And so every substance that is in blood gets filtered and at some point ends up in urine, including HCG. Okay? So if you pee on that stick and you see a smiley face or a plus or a frowny face, however they designed it, okay, that means that there is HCG in your urine. That means that somewhere in your body there's an embryo, presumably the uterus, right? And hopefully it's not something else, that produced HCG, and so you're pregnant. Make sense? There's other ways that scientists try to figure out how to develop a pregnancy test. So antibodies were only around since the 1970s, okay? So the other ways that this was sometimes done or kind of experimented with is with frogs. We all know what a frog looks like, right? You know a little bit about the biology of a frog? So do frogs lay eggs or do they give live birth? Yeah, they do eggs. Very good. Awesome. Okay. So, and not just one egg, there's a crap load of jelly-like stuff. Okay. So what do you think would happen if you have a female frog and you inject it with urine from a pregnant woman? So you're holding that frog in your hand, you have a needle, it's loaded with urine from a pregnant female. Frog's trying to get away, but you have a good grip on it. What's going to happen? Yeah, but what physically is going to happen? It's going to dump out a bunch of eggs. Okay, so it releases eggs. Okay, how? Why? How? Because it's so freaked out because of that needle? So what is it that causes that to happen? HCG. So what is happening to that frog? Basically, what are we mimicking? Not fertilization. We're not fertilizing. I haven't even talked about sperm yet. I didn't talk about a male frog. There's no sperm. Ovulation, exactly. Remember what I said? HCG is just like LH. If you inject that and there's lots of HCG, you pretty much caused an LH surge. Okay, so that frog sees lots of LH and starts ovulating. Okay, now that would be an interesting pregnancy test the next time you go to Walgreens, right? Little jars of frogs that are sitting there with a needle attached to it. Okay, let's move on. Okay, so again, the key thing here is that we have to maintain high levels of progesterone and estrogen throughout pregnancy. So throughout these 9 months, estrogen and progesterone levels need to remain high. Okay, and I think I've mentioned that at least a dozen times now. If that doesn't happen, the endometrial lining sloughs off. You cannot maintain pregnancy. Okay, so early on, the estrogen and progesterone comes from the corpus luteum. If by the time we have a blastocyst, an implantation, the trophectoderm secretes HCG, lots of HCG, so we keep the corpus luteum around. Continues to secrete progesterone and estrogen. What happens to the trophectoderm at some point? Yeah, it turns into a placenta. Okay, so we keep the corpus luteum around, produce progesterone and estrogen. The trophectoderm, in the meantime, develops and differentiates into a placenta, and the placenta now, instead of continuously secreting high levels of HCG, will just secrete estrogen and progesterone itself. Okay, so for most of the pregnancy, that estrogen and progesterone comes from the placenta. Okay, so as soon as we have a placenta, all this stuff comes from the placenta. Okay, and then at some point, a little after 9 months or 9 months, all of a sudden we see this gigantic drop in estrogen and progesterone. How is that possible? Yeah, so this is upon delivery, right? And obviously with delivery, we don't only deliver a baby, we also deliver a placenta. The placenta leaves as well. If the placenta is out of mom's body, all that estrogen and progesterone is gone, it drops, and what happens? We can start a new cycle if we want. Okay, what are we doing with the placenta? Eating it, right? No, you can, right? Some people, some cultures, people decide to eat it. We now have companies that will pulverize it for you and heat treat it and put it in a capsule. So you can take the capsules and that way eat your placenta. Some people take it home and plant a tree on it. Are there any health benefits to eating a placenta? Yes, but have you ever heard of something called a placebo effect? That's exactly what this is. Think a little bit about what a placenta is. By the time we talk about encapsulating placenta in pills that have been pulverized and heat inactivated and stuff like that, There is no nutritional value to that stuff at all, except for if you just think hard enough that it makes you feel good, it will, okay? I have no idea. If you take a placenta, you dig a hole in your yard and you plant a tree on it, I have no idea if that tree grows because of the placenta or not, okay? You will have to plant 2 trees right next to each other on opposite ends of the yard and have one with a placenta underneath it and the other one without. This is more historically because what does the placenta do? Provides life, fetal growth, okay? It's kind of a sign of promoting life, okay? So it has more of that stuff than physiologically providing any benefits. This is a mass of tissue, highly vascularized, lots of blood, and lots of hormones. That's it, okay? Okay, let's talk about a placenta. I really wish I had a lot more time to talk about a placenta because it's a fascinating organ. There's lots about the placenta we could talk about. All I want you to recognize here that now we talk about a fully differentiated placenta that all started from that single cell layer that was the trophectoderm, that single layer, okay? That is going to turn into what is kind of like pink here, which is now called not trophectoderm anymore, but we call it a chorion. Okay, so the chorion is the fetal part of the placenta. Okay, because with the placenta there's a maternal part and there's a fetal part. The chorion comes from the trophectoderm, from the embryo. Okay, and then you see these tree-like structures. Those are called chorionic villi. Okay, now in terms of a function of the placenta, the 2 main functions really are be a semi-permeable membrane that allows oxygen and nutrients to go to the fetus, and carbon dioxide and waste to leave the fetus and go back to mom. Okay. The second thing that the placenta does, we already talked about, estrogen, progesterone. It is a huge endocrine organ. It secretes lots of these hormones, and it's necessary for pregnancy. Okay. So those are 2 very important functions of the placenta. The second thing we have to recognize is that because of the beautiful thing called meiosis, however, now because of meiosis, we have created an individual that is genetically not identical to mom. So we can absolutely not have fetal blood and mom's blood mixing. It has to be separated. Okay? If that were to mix, then all of a sudden it will reject the pregnancy, or the fetus, sorry. It will reject the fetus. Okay? So when we think about the placenta and how it's set up in humans, really what we have is these chorionic villi that are highly vascularized, and these, the vasculature here is covered by a really thin cell layer, okay, a really thin cell layer. And that thin cell layer separates fetal blood, which is within the chorionic villi, and maternal blood, which is all this maroon colored stuff that the chorionic villi are bathing in, okay. So there's a very thin cell layer that separates fetal blood from maternal blood, and we can absolutely have no mixing of blood. Okay? Make sense? The final thing I want you to know about the placenta is that blood flow is a little odd, because in this case we have umbilical vein carrying oxygenated and nutrient-rich blood to the fetus, whereas the umbilical artery takes carbon dioxide and waste, carries it from the fetus to back to mom. Okay? So in this case we have oxygen-rich blood in umbilical veins and not in umbilical arteries. Okay. Any question about placenta? Yes? Stem cells in the umbilical cord. You're absolutely right. You have something called umbilical cord bank or banking. Okay, because just like the placenta, you could take it home, or if nobody wants to take it home, then they just throw it in the trash. Really, that's how it works. The same thing with the umbilical cord, okay? You get to cut it, and after that, you know, it is what it is. There are mesenchymal stem cells in the umbilical cord that people store and then hopefully could help in terms if down the road that baby needs certain stem cell treatments, okay? Now, how well that really works or not, I'm not really sure, okay? Because mesenchymal stem cells are not like embryonic stem cells. Embryonic stem cells really can give rise to everything that you can possibly think of except for the placenta. Mesenchymal stem cells are definitely stem cells, but they don't have the same type of potency in terms of all the different cell types they can give rise to. Okay? So you definitely could culture those cells, you could coax them a little bit to help differentiate in certain things, but I'm not sure in terms of the breadth of application that it could be used for. But lots of people do it. Okay, so that's definitely another option. I'm sure, considering the US is a capitalistic economy, there is definitely lots of opportunities for companies to really promote this as well. It's not cheap. Nothing is for free in the United States, including this education, right? Okay, so pregnancy, okay, lasts for 9 months normally, okay, and then we get childbirth, so parturition. So we have to talk about contraction, okay? So basically what happens is throughout the 9 months of pregnancy, the fetus is getting bigger and bigger, it continues to grow, and as such, it's also going to drop lower and lower into the uterus, okay? And at some point what's going to happen is when it drops lower onto the uterus is that it pushes on the cervix and it causes cervical stretch. Sensory receptors in the cervix obviously notice this and they signal to the brain. And specifically, it acts on the posterior pituitary gland to secrete oxytocin, okay? So oxytocin, a hormone from the posterior pituitary gland. There's another hormone from the posterior pituitary gland that you guys know, since you're all experts on kidney physiology. Vasopressin, right? ADH, vasopressin, also posterior pituitary gland. Oxytocin is the other one. Oxytocin into the blood, goes back to the uterus, causes uterine contraction. So it acts on the myometrium, causes the muscles to contract, okay? In addition, oxytocin also acts on the uterine wall to cause the release of prostaglandins. Prostaglandins also cause the uterus or the myometrium to contract. If you contract the uterus, what's going to happen to that fetus? You're going to push it more on the cervix, right? It's going to drop even lower. So that's going to increase cervical stretch. So basically what you have here is a whole bunch of positive feed-forward loops that cause more regular and more intense uterine contractions, okay? Now, one thing that I quickly want to mention is, and I'll get back to this slide in a second, is going back to this graph, okay? At one point, I put a slide on that talked about estrogen and progesterone. One of the functions of progesterone was it inhibits myometrial contractions. Now, if you look at this graph, progesterone is always higher than estrogen. So how do we deal with this? We have progesterone levels so high that inhibits myometrial contraction. Well, the one thing that it works is we get something called functional progesterone, or P4, withdrawal. Okay, functional progesterone withdrawal. Okay. What that means is there's always lots of progesterone. However, you guys all know that progesterone is a steroid hormone, and the only way it functions is through its receptors, progesterone receptor. And there's different isoforms of progesterone receptor, but basically what happens near the end of pregnancy is that the receptors are not working the same anymore. And think of them as they're just not functioning at all anymore, almost. Okay? So even though you have lots of progesterone around, if the receptor doesn't function as well, okay, then we don't have that inhibition to myometrial contractions anymore. And in fact, functionally speaking, we still have lots of estrogen, still lots of estrogen receptor. So estrogen actually starts becoming more dominant in its function by stimulating myometrial contraction. Okay, does that kind of make sense? Does it make sense what functional progesterone withdrawal means? So just remember, progesterone is always high, and it will stay high. It doesn't disappear. It has to do with the receptor for progesterone. That's it. Okay? So what I want you to do on this figure right here, to kind of have that complete picture of parturition, what you can do is put estrogen right here on the bottom and an arrow towards uterine wall prostaglandins. Okay? So estrogens then, because they're always there too, from the placenta, okay, acts on the uterine wall, causes release of prostaglandins. Progesterone is not really inhibiting this anymore, okay, and you get continuing uterine contraction. Okay? Okay, and then we have a baby. Last thing is lactation, okay? So we're going to talk about hormones again, okay? And I think this is the last hormone that we're going to introduce And that's it. And so basically what I could do is just have 100 questions about hormones, but that's not what the exam is going to look like. Don't worry. Puberty, estrogen and progesterone are important, okay? So estrogen, in fact, during puberty increases duct growth and branching, whereas progesterone promotes alveoli growth. So breast development during puberty, we already know that, okay? What happens during pregnancy is estrogen and progesterone also promote breast enlargement, okay? And prolactin is important for milk production, okay? And even though there is prolactin, okay, that is involved with breast enlargement as well, there is no milk production. And the reason for that is that prolactin comes from the anterior pituitary gland, okay? And what happens with estrogen and progesterone, well, what does it do to the brain? Luteal face. What does estrogen and progesterone levels do to the brain? Inhibitory, okay? So it shuts the brain off in terms of hormone release, okay? So basically, estrogen and progesterone levels, as they're really high during pregnancy, inhibit the release of prolactin, okay? What happens once the placenta is gone? Estrogen levels and progesterone levels drop, and so now we have lots of prolactin. And we get milk production, okay? So that's only 1/2, because that talks about milk production, okay? We still need to talk about milk ejection, okay? So lactation, milk secretion. So what I just talked about is kind of on the left side here, and what I want you to do is take a marker and cross out this little part right here about dopamine and PRF. PRF is supposed to stand for prolactin releasing factor, but since we don't really even know what that does or is, just ignore that. So just focus on this part right here. So we just talked about that prolactin causes milk production. The stimulus is really the baby nursing or crying, anything that could act on the brain that causes release, that signals to the anterior pituitary to release prolactin, gland cell stimulation, increase milk production. At the same time, these same neuronal inputs act on the posterior pituitary gland. That causes release of oxytocin. Just like oxytocin promotes uterine contractions, it also causes contraction of myoepithelial cells that allow milk ejection, okay? So for lactation, all you have to know is that anterior pituitary gland secretes prolactin, causes milk production. Posterior pituitary gland releases oxytocin, causes milk ejection. That's it. Make sense? Awesome. Okay, now the last part. Applied reproductive physiology. You are all experts now. We're now going to pretend we are all MDs in a fertility clinic and see how far we get, okay? So the one thing I want you to recognize, even though we're going to spend most of our time talking about IVF, in vitro fertilization, assisted reproductive technologies is more than just that, okay? Women undergoing hormone treatment to regulate their monthly cycles, that is technically also assisted reproductive technology, okay? We'll talk about hormones as well with IVF, but anything that a couple does related to hormone injection, sperm transfer, okay, IVF, all of that stuff is assisted reproductive technology. We won't really talk about stem cells or cloning too much, so you can ignore that. We'll talk about only this cloning and somatic cell nuclear transfer. That's the other thing we'll talk about, okay? And then we'll finish up with the real reason why everybody is here, because the exam questions, which is good. Okay, fertility. We've asked this question before. What happens to fertility with age? It goes down. So nothing shocking, right? And so this is focusing on maternal age here. We also mentioned that paternal age is also not great because you accumulate mutations and the chances of an egg being fertilized by a sperm-carrying mutation increases with age. And especially things like autism and some neurological disorders is definitely linked to advanced paternal age. Here we're showing that maternal age, with increase, we see a decrease in fertility and increase in spontaneous abortion. Okay? And all we've talked about so far is the fact that maybe it made sense a little bit if you think about the oocyte and what the oocyte is asked to do. okay, completing meiosis. When it ovulates at the age of 20, it's been arrested in meiosis 1 for 20 years. If it ovulates at the age of 50, it has been arrested at that stage for 50 years. And when we do these meiotic divisions, these chromosomes need to separate properly. Okay? So maybe it's not too surprising at all that this is the case. But like we said the other day too, everybody already knew this. You've heard the term biological clock already, but keep in mind that when you're a 40-year-old woman, you could still have a baby, a healthy baby, okay? This is not all or nothing. There's not all of a sudden, from here we go to no babies anymore, okay? Okay, so let's focus on IVF a little bit. Does anyone here know the name of the first IVF baby? Any idea? Louisa Brown. Louisa Brown is the first IVF baby, okay? When do you think the first IVF baby was born? A rough guess. 5 years ago, 10 years ago?
1975.
Oh, very close. 1978. Okay. This may be a good day for you to take the exam. I mean, we're pretty close, okay? 1978. This is John and Leslie Brown with their baby, Louisa. This was the headline in a newspaper. Okay, me, Louisa, the world's first test tube arrival super babe. Okay, so clearly when we look at that kind of wording, we don't call these babies test tube babies. It's a very unpleasant, unfair term at all. If anything, it's wrong because it's not made in a tube. So if you want to be very practical about this. So I really want to focus on the IVF procedure itself, and I don't want to get sidetracked too much about you know, the ethical things about IVF. How many clinics do you think there are in the United States right now, fertility clinics that do IVF?
20,000.
No, that would be a lot. It's a little less. According to the CDC, in 2014, 4 years ago, 498 clinics. Okay, and that has only increased from there on. So the other thing is, if let's see, what was the other? So in 1978, since 1978, okay, 5 million babies have been born worldwide since through IVF. Okay. In 2014, 498 clinics. At that point, there had been 208,000 ART cycles performed. That's almost 250,000, again, 4 years ago. Okay, so there is nothing wrong with IVF. Just like adopting a baby is an opportunity for a couple to have offspring, IVF is an opportunity and a possibility for a couple that is infertile to have offspring. Okay. And I really kind of want to focus more on the reproductive biology part of this. Okay, so clearly a technique that is used a lot, leads to birth of healthy babies. Louisa Brown is 40, she's turning 40 this year. Okay, there's nothing wrong with her. She has 2 kids of her own. Okay, not through IVF or ART, just normally. Okay, so great opportunity. Okay. Now, I want to focus on the IVF procedure, and since we're all experts now anyway, you guys can help me out a little bit here. In vitro fertilization. The first thing we're going to do is we're going to stimulate egg production. So that means that we have to inject the woman with a hormone. Okay? What hormone are you guys going to inject into that woman? So progesterone, estrogen, LH? That's the one. How many think LH? FSH? I'm going to pick FSH, which is really not fair because I know the answer anyway. What does FSH stand for?
Follicle stimulating hormone.
Voila, follicle stimulating hormone. You stimulate egg production, you want to inject FSH, follicle stimulating hormone. Okay? And basically, that doesn't happen really in a clinic. The women can do that at home. Okay? Basically, what that does is we're stimulating lots of follicles. Okay? We normally already have a group of follicles, but we're really going to promote 10 to 20 follicles to continue growing and not dying off. Okay? So we make sure there's enough FSH around to support lots of these larger follicles. Okay. Now, when we do this and time this correctly, how do we get these eggs out of the ovary? In the previous group, somebody said surgery, which, sure, but there's something maybe less invasive. How do eggs normally get out of an ovary? Through the fallopian tube, but yeah, but what do you do hormone-wise? What hormone causes eggs to pop out of the ovary? Ah, there we go, LH, there we go, LH. LH causes ovulation, okay? So LH, you can provide LH. We talked about another hormone today that is similar to LH that you could then also inject that does the same thing, which is what? HCG, human chorionic gonadotropin, right? So both of that, now all of a sudden we have a dish and it contains lots of eggs, okay? So now we need, that is 1/2, obviously we need to have a contribution from the male as well, okay? So what hormone are we going to inject into the male to get sperm? Estrogen? Men don't like estrogen. It doesn't work well for us. Testosterone? Anyone?
Too much test causes sperm count to die.
Yeah, so testosterone probably not good either. So for all the men that are in this room, okay, if you ever end up in a fertility clinic and they want to inject you with something to get sperm, you need to walk away or run away, okay? All men need, and you guys know this, maybe a quiet room, a magazine, and some kind of a video. That's how we get sperm. Unless there is a blocked urethra, okay, sperm will come out. Whether it's normal or not, it will come out, okay? We don't need hormonal injections, okay? So now we have a dish. We have eggs. We have sperm. We're going to look under a microscope. What are we going to look for under a microscope?
Good swimmer.
Good swimmers, that will help, right? The ones that don't swim as well, they'll just stay. Those are crappy anyway, right? How can we actually check and see if an egg is fertilized? What are we looking for? 2 polar bodies and 2 pronuclei, okay? That is something you could see under the microscope. Then you can actually take those, you put them in a separate dish, and you can allow those to develop. That's going to go turn into a 2-cell, 4-cell, morula, and then when it's a blastocyst, then you can transfer them back into mom, okay? So the blastocyst is transferred back into mom. How many are we going to put back into mom? And before answering the question, I want you to recognize 2 things. How expensive is this? Yes, $15,000 to $20,000, okay? Number 2, how efficient is this, you think, in terms of having a live baby being born? 80%, 50? 29 to 30%, okay? Now, let's go back to that question that I asked. How many do we want to put back? 15. Make sure you get some. 2 to 3. You're absolutely right. Okay, 2 to 3. Now, can you do more? Yeah, for sure. Okay, and you guys already know this. Have you ever heard of quadruplets? Octuplets? Okay, so most clinics will do 2 to 3, okay? But you have to recognize that, again, this is a capitalistic economy, and fertility clinics are not sponsored or get funding from the government. They make money. The way they make money is have lots of clients. The way they get lots of clients, if it's you as a couple know that the chances of me getting a baby is very high. Now, if you only transfer 1, that's an issue, right? Because you're more likely not to have a baby. If you have 2 or 3, yeah, chances increase a little bit. But the chances also increase of getting twins and triplets, right? So, kind of tricky. The US, the government does not regulate this, okay? This is not regulated by law. In the United Kingdom, it's a little different. It is regulated by law. And some of the factors that they look at is maternal age. So, for example, only if mom is older than 40 years of age will they transfer 3 or 4, okay? If it's a 20, 25-year-old individual, they're not going to transfer 3 or 4. They may only transfer 1 or 2. And that's regulated by law outside of the United States, okay? Okay, that's it for IVF. Now, there is something called ICSI, intracytoplasmic sperm injection, which is very similar to this. The only difference there is you take the sperm and you inject it into the egg. And in fact, most of the cycles that are happening right now are probably more ICSI than IVF. Okay, so now you just take the sperm, inject it into the egg, and let that develop. Okay. Okay, any questions about ART, IVF? All good? Okay, just one quick slide about cloning. Okay, reproductive cloning. So reproductive cloning, when we talk about cloning, we often think about reproductive cloning, which is basically making a copy of yourself. Okay. So what is on the cover of Time magazine here? Who's that? Dolly. Okay, we all know Dolly, and Dolly was named after Dolly Parton. And the reason she was named after Dolly Parton is because of?
Mammary stem cells.
Yes, no, mammary gland, not mammary stem cells. Okay, mammary glands. Apparently, Dolly Parton had big mammary glands. Okay, Dolly, the sheep, does not have big mammary glands. There's nothing to do with that. In order for this to make sense, we need to think about this process, highlighted in red. So I want you to recognize that process, which is called somatic cell nuclear transfer. And that is basically a technique to do cloning, or reproductive cloning. What you do here is you take an egg and you remove the chromosomes. You get the nucleus out, okay? Often referred to as an empty egg, which is kind of bad because there's still mitochondria and all that stuff, but the chromosomes are gone. Then you take a somatic cell, any somatic cell, it doesn't matter, and you take that nucleus, or you fuse that cell with the egg. But basically, you want the nucleus or the DNA from that somatic cell. In the case of Dolly, exactly, it was a mammary gland cell. It wasn't a stem cell, but it was a cell from a mammary gland, okay? And how many chromosomes, let's pretend sheep have the same number of chromosomes as we do. How many chromosomes are in a mammary gland cell? 46. So you put 46 chromosomes in that egg, okay? And then it's really kind of like the Frankenstein movies where you add a little bit of electricity, which kickstarts embryogenesis, okay? And once embryogenesis starts, it's blastocyst, and that's what you transfer back into mom, okay? So that is the whole process of somatic cell nuclear transfer. When you do that, that's how you clone animals, okay? Theoretically, that's how you could clone humans, but that's horrendously stupid because of several things, in addition to ethical reasons, okay? How efficient do you think this process is? Like 10%? It's actually 0.3%. So just think about the number of eggs you would need in order to get one offspring. Okay, so you can already not ask women to donate eggs. Like, I cannot give you, what, $10,000 to give me some of your eggs? That's illegal, okay? And in addition, there is no purpose to reproductive cloning, okay? Because reproductive cloning, just like identical twins, which are clones from each other, depending on where they grow up, they may get different diseases. One may be good at soccer, the other one may be good at something else. One may be smart, the other one is stupid. I mean, there's lots of other things that impact phenotype and certain abilities or traits, okay? So reproductive cloning is not interesting. Recognize here that obviously this is kind of stupid. This should be a sheep, okay? That's kind of how somatic cell nuclear transfer works, okay? Okay, so the reason why this procedure is of interest, which you don't have to write this down or anything, is the therapeutic cloning. Not a reproductive cloning, but the therapeutic cloning. Because if you could do this stuff, take a skin cell, take the nucleus, okay, put it into an empty egg, allow that to develop as an embryo, boy, now I can grab those inner cell mass cells or these embryonic stem cells, and now I have embryonic stem cells made from my own skin cells. So basically, you asked about stem cells in the umbilical cord. Well, that's fine and dandy, but this way allows me to get embryonic stem cells for just me, okay? Again, the problem here is ethical reasons because you're destroying embryos. And again, that's not allowed, okay? Okay, but there is definitely potential or an interest in terms of treatment for diseases such as Parkinson's disease, Alzheimer's, things like that. Can we fix certain neurons? Can we regenerate beta cells with type 1 or type 2 diabetes, stuff like that. Okay, any questions about this stuff? All good? Okay, so finally, here we are. Exam questions. Okay, so just a short question. Which of the following is not involved in primary sex determination? A, B, C, D, or E? B, right? You don't, I'll post it, no worries. I'll post this stuff. I mean, go ahead, you can take pictures too. It's all fine, but I'll post it. This is a very wordy one, so I'm sorry. Just, this is kind of, I need to come up with something, okay? So, correct order of germ cell differentiation in adult testes. E is kind of starts right here. So it's probably not E. Boy, we're starting to feel awfully good about this final exam, right? Awesome. So this is kind of a horror scenario for a man that goes to one of those horror IVF clinics. But let's just presume we need to get sperm. Which one can we use from this to do IVF or fertilize an egg in vitro? So now there's a whole bunch of maybe angry women that chase a man with a needle and want to collect sperm. So now think again about something about maturation of sperm. Not capacitation, that's in the female reproductive tract, but some other stuff. So which one? E, the reason for that is it acquires the ability to become motile. It will not be motile, but boy, if you take it out and put it in a dish, it can become motile, but only by the time it is in the tail of the epididymis. Okay? Good luck. Have fun. Good luck on the exam. Don't worry, it's not the end of the world. It's going to be okay.